Protozoa. The word means first animal. The protozoa are considered to be a subkingdom of the kingdom Protista. More than 50,000 species have been described, most of which are free-living organisms. Your protozoa are found in almost every possible habitat. These organisms are unicellular, but are animal-like in that they are motile, and some are capable of ingesting or eating their nutrients, not just absorbing them. Virtually all humans have had protozoa living in or on their body at some time, and many people are infected with one or more species throughout their life. Some species are part of the normal flora. They're not normally harmful, whereas others are pathogenic and usually produce disease. Protozoal diseases can range from very mild to life-threatening. Although protozoa don't play a significant role in the health of humans in developed countries like the US, today, I expect their impact will increase as climate change progresses in upcoming years. Your objectives for this video are to list some basic characteristics of protozoa. You should be able to distinguish between trophozoites and cysts, and you should be able to describe how protozoal infections are acquired and diagnosed. Finally, you should be able to describe some common protozoal diseases in the US. So, protozoa are a diverse group of organisms. As you can see from this picture here, they have a diverse set of shapes. They can be ovals, they can be trumpet-shaped. And some of them, like this amoeba down here, look like a paint splatter. They live in a variety of aquatic environments as well. They can be found swimming in fresh water or saline. They swim and crawl along tissues, and they can be found in decaying leaf matter. They just need a little bit of water. Your protozoa are motile. Remember, that means that they can move from one place to another under their own power. And there's only a few that aren't able to do this. Those non-motile protozoa tend to be obligate intracellular parasites, and they belong to a phylum called Apicomplexa. Your protozoa have two basic forms. You have the trophozoite form. Can you say trophozoite? Trophozoite. So the trophozoite form is the form where the protozoan is actively absorbing or ingesting nutrients and growing. You can think of this as being the feeding stage or the feeding form. Alternatively, the cyst form is the hardy, dormant form of the protozoan. It's also the infectious form. So under unfavorable conditions, a trophozoite may encase itself in a cyst, which protects it from environmental insults. These cysts can be released into the environment and persist for several weeks to more than 20 years under favorable conditions. Alternatively, some protozoa form cysts in tissues, not just out in the environment. These cysts will have a thinner cell wall than the ones that are out in the environment. When cysts are ingested via contaminated food, beverages, or fingers, the cysts travel through the upper digestive tract. They go through the stomach and they're protected by that cell wall. In the small intestine, the trophozoite will crack open the cyst wall and emerge to start an infection. Protozoa are almost always in a hypotonic environment. So that means that they're at risk of osmotic lysis. Instead of making a cell wall, protozoa have a firm but flexible covering called a pellicle, Pellicle, and that reduces how much they can expand when water rushes into them. Some may also have a contractile vacuole that acts like a pump to pump water out of the cytoplasm. As eukaryotes, protozoa are capable of asexual reproduction and sexual reproduction, and we're going to look at these in a little bit more detail shortly. Asexual reproduction is by fission, so the protozoan splits itself in half, budding, which is similar to what we saw with the fungi, or a special protozoal form of asexual reproduction called schizogony. Schizogony. Sexual reproduction is by conjugation, and this is not quite the same conjugation that we saw back when we were talking about prokaryotes. So one of the unique characteristics of your protozoa is their methods of motility. Your protozoa will all use one of three methods of motility. The first is called a pseudopod. The plural is pseudopodia. Now this comes from the Latin. Pseudo means false, and pod here refers to feet. Think about like podiatrist or podiatry. You know, that's like a foot doctor or the study of feet. So that's the same root as the pod here. So what happens with these protozoa is they essentially move their cytoplasm in order to move the cell. So they will send a projection out like this one into the environment ahead of the cell, and then they'll move the cytoplasm, the rest of the cytoplasm, towards that point. And they can they can make these pseudopods, or they can retract them. So they can move out away from the cell or move back towards the cell. It just depends on the situation. And this gives these amoeboid protozoa kind of a creeping action, so they creep along surfaces to get from one location to another. The reason why this happens is because of cytoplasmic streaming. So there are proteins, cytoskeletal proteins, that are part of the cytoplasm for these cells. And these proteins are called actin microfilaments. When an amoeba wants to move, it basically stacks actin monomers on top of each other to make a filament. And then it takes them apart one by one. And so it makes a pseudopod by adding the monomers together to make a filament, and then to move the rest of the cytoplasm towards that projection, that pseudopod, it breaks the actin filaments down. Now I don't need you to know all of the details of that, but you should know that a pseudopod is a projection that's made by amoeboid protozoa, and it's used for movement. Another structure that's used for movement by some protozoa are the cilia. Now you've seen these before, right? These are short hair-like structures that are made up of microtubules and motor proteins, and they beat rhythmically. And protozoa use this beating movement to propel themselves from one place to another, or to move food that is nearby towards their mouths. Protozoa also have flagella. These are eukaryotic flagella that are also made up of microtubules and motor proteins. The protozoa can whip these flagella back and forth and use that to move themselves from one place to another. So here are some examples. This is Giardia lamblia, and these hair-like structures that you see, those are flagella. This is a flagellum, kind of like an endoflagellum, that's on another protozoan called Trypanosoma brucei. I've put a video on motility in your lecture videos folder. So it basically describes these structures in a tiny bit more detail, but it has the added benefit of showing you examples of protozoa moving using each of these structures. I highly recommend watching the video. So now let's talk about reproduction, and we're going to use amoeba as an example here. One thing to know about your protozoa is that they have two nuclei, a micronucleus, which is responsible for transmitting genetic information from one cell to another, and then they have the macronucleus, which basically is made from the micronucleus, but It contains RNA as well as DNA, and it's responsible for running the daily functions of the cell. So the micronucleus just transfers DNA, the macronucleus controls the cell. All right, now let's take a look at what happens. In this example, we have a fully grown amoeba, and it's going to undergo reproduction by binary fission on the left side of the figure. This starts when the amoeba retracts all of its pseudopods and becomes more rounded and ball-like. Then the micronucleus divides by mitosis, producing two new micronuclei. Then the cell constricts in the middle. You probably remember this from Gen Bio, and splits. Okay, from the micronucleus, we'll get a new macronucleus, and that's what the green thing is here. That's supposed to be the macronucleus. And now we have two daughter cells, two new amoebas. So the cell just split itself in half. Multiple fission here on the right is another name for schizogony. And we see this type of reproduction in parasitic protozoa called sporozoa. Basically what's going to happen is that our amoeba again retracts all of its pseudopods and it begins to round itself up. Then the amoeba develops a cyst cell wall. Now this is going to happen under unfavorable conditions for this amoeba's growth. It's ready to go dormant, and it's going to hang out in this dormant form until favorable conditions resume. Then a bunch of new amoebas are going to emerge from this cyst. That's the big view here. So where we left off, the amoeba was secreting its cyst wall, which is going to protect its nucleus, its cytoplasm, etc. Inside of this cyst, the micronucleus undergoes tens of mitotic divisions. So you could have 50 of these inside of the cyst, 50 nuclei. Then cytoplasm from this amoeba, from this cyst, kind of condenses around the nucleus, and you get the formation of plasma membranes. And the cell, or the cyst rather, will exist like this. When conditions are favorable for growth of the amoeba, the cyst will rupture, releasing new amoebas. And again, you could have tens of amoebas being released from this one structure. Now you might be wondering, okay, who cares? That's an amoeba. Why should I know this? The reason why you should know this is because there are some really dangerous protozoa that use schizogony to increase their numbers during an infection. And one such protozoan that does this is called Plasmodium falciparum. It's the parasite that causes malaria. So now would be a good time to pause this video and watch the videos in your lecture videos folder that are in the malaria folder. So inside the lecture videos folder, there's another folder that says malaria. Watch the videos in there. It goes through the life cycle for this protozoan. Then join me back after you've seen that video. So now you should have an idea of how malaria reproduces both in humans and in mosquitoes. And what I want you to be aware of is that you actually saw schizogony happen. In the part of the video where the parasite infected a human being and went into the liver and infected liver cells, inside that liver cell, schizogony happens. And when that liver cell is full of parasite, then it ruptures, releasing those parasites into the liver. And then they find their way into the blood, right? And then they travel through the rest of the body. So in the liver cell is the first schizogony event that you saw. The second time you saw schizogony was when the parasite infected a red blood cell. In the red blood cell, the parasite reproduces tens of times, producing lots and lots of new parasite. And then they rupture the red blood cell. So that was the second schizogony event that you saw. And so you can imagine how you gradually increase the number of parasites that are inside of the body really quickly, because each time that a cell becomes infected, you release 30 to 50 new protozoa. Okay, so that's not trivial. The third time that you saw schizogony was actually when the parasite was inside of its mosquito host. So you get the formation of an oocyst, like a fertilized egg. Okay, inside of there you get the a schizogony event, and when that ruptures, you get the release of a lot of get the release of a lot of parasites, and those parasites then go and infect the saliva of the mosquito, completing the life cycle, right? Because when the mosquito goes and bites a new human host, some of those parasites get spit out, expectorated into the wound and into the new host. So you saw schizogony 3 times. Sexual reproduction in protozoa is called conjugation, and it's very different from anything you've ever seen. Really briefly, let me describe what happens. So we have 2 protozoa here, paramecia. Okay, the large oval that you see, that's the macronucleus. Remember, that's the one that runs the function of the cell. The little tiny one, the little tiny circle, that's the micronucleus. Remember, it transfers genetic information. So the first thing that happens during conjugation is 2 different protozoa come close together, close enough to exchange DNA. When that happens, the micronuclei begin to reproduce via mitosis, producing lots and lots of copies. Now, some of these copies will be identical to each other. So in step number 3, the identical copies will actually get destroyed. They'll degrade. Step number 4, you get the exchange of a micronucleus between both organisms, and then the organisms detach here. So now you can see that they have 2 different micronuclei inside of them. Okay, these will fuse together, forming a synkaryon. Okay, and then that synkaryon reproduces several times, producing lots and lots of copies. Next, the macronucleus disintegrates, and you get the formation of new macronuclei, okay, from some of the micronuclei in step 7. Then you get the splitting of the cell to produce new protozoa. So that's kind of how conjugation happens. I don't necessarily expect you to remember every single step, but you should have the general idea that there's going to be an exchange of DNA. The cells are going to get close together. There's going to be an exchange of micronuclei, which is where the gene mixing comes. Okay, then those micronuclei fuse together. We're going to get duplication of the fused micronucleus. That was called the synkaryon. And then we're going to get macronuclei developing from those micronuclei. And then we'll get new protozoa. So now I want to wrap up this video by talking about some diseases that are here in the United States and are caused by protozoa. Humans and animals become infected by protozoa when they, for example, eat contaminated food. It could be that the food was washed in water that contained cysts, or maybe it's a salad and the field where the lettuce was growing was irrigated with water that had cysts in it. Maybe it's that the person who's preparing the food didn't wash their hands after using the restroom, and their hands have feces on them, and they've been preparing food for someone else. Then the food becomes contaminated. There's a number of ways that it can happen. You can also become infected by drinking contaminated water. Again, not that uncommon for water out in nature to be contaminated with cyst forms of protozoa. Sometimes people are infected when and animals are infected when they're bitten by infected insects like mosquitoes, ticks, tsetse flies, and other types of insects. There's the fecal-oral route as well. When I think about fecal-oral route, again, that person who didn't wash their hands after the restroom would be an example of that. But also children, potty training children, toddlers, they may not wash their hands very well after reaching in a diaper or using the potty, and then they go touch toys that other kids put in their mouth, and that can transmit protozoal infections. In fact, for some of the diseases that we'll talk about, kids are more likely to get the disease than adults for that very reason. Then, of course, if you eat food with contaminated fingers, you can become infected. Many of the protozoal infections that we'll talk about today are diagnosed by what's called a stool O and P. So a stool O and P, the O stands for ova and P stands for parasite. Basically, what happens is you take a fecal specimen from someone, dilute it a bit, smear it on a slide, and then prepare it for viewing under the microscope. And you look for eggs in the case of parasitic helminths, or you look for parasites like these protozoa that I'm talking about. Okay. There are 8 different phyla of protozoa. 4 of them contain human pathogens. I've listed them here. Of these 4, probably the most important is Apicomplexa because it contains a lot of parasitic protozoa that are responsible for some pretty significant human disease. All right, let's talk about our first disease. This is Giardia. It's also called beaver fever because there was an outbreak in which hikers at Banff National Park became ill from drinking stream water contaminated with Giardia from beavers. So the beavers had basically defecated cysts into the water, and then these hikers were downstream and drank some of that water. Yeah. Okay, so anyway, beaver fever. Basically, this is caused by a protozoan named Giardia duodenalis. In the past, it's also been called Giardia lamblia or Giardia intestinalis. So these are all referring to the same protozoan. And it's the most common protozoan in stool specimens in the United States. This protozoa tends to be found more often in children, especially young children or toilet training children, but there are lots of adults who get infected as well. Giardia is a leading cause of waterborne outbreaks of diarrhea. It causes about 1.2 million incidences of illness in the United States annually. Okay. Typically, people become infected by drinking contaminated water. So the water has cysts in it. And I'll be honest, the occurrences are more common in the western mountain regions of the U.S., like around the Sierra Nevadas, the Rocky Mountains, and the Cascades, because people will be hiking around in the mountains, they'll get thirsty, they get water from a stream or a lake, and they don't filter it properly, and then they consume cysts. Okay. That said, the parasite can also be found in municipal water supplies sometimes, swimming pools, whirlpools, spas, and wells. Giardia can also be transmitted through food. So, someone, again, has been irrigating their crops with contaminated water, or they've washed food with contaminated water. Okay, that's a source of infection. It's also transmitted by person-to-person contact. So in this case, someone comes in contact with someone else's fecal matter. Okay. All right. So here's what typically happens. The person drinks cysts in contaminated water. In this cyst form, the parasite is able to withstand the upper digestive tract. It makes it through the stomach and goes into the intestines. Then the trophozoite emerges from the cyst, and it's going to attach to the intestinal wall. This is the small intestine. Okay, so it attaches to the intestinal wall through this sucker mechanism that you see right here. Okay. All right. So while it's attached to the tissues, to the intestinal wall, it starts reproducing and absorbing nutrients from the lumen of the small intestine. And for most people, the parasite just kind of stays there. Okay. Now, some people who are infected with Giardia don't know it. They experience no signs or symptoms. I don't know if we've talked about this before, but a sign is something that can be measured or observed. Symptoms are things that are felt by a patient. So, for example, a sign might be fever because you can measure that, swellings because you can see that, um, increased heart rate because you can hear that with a stethoscope, right? A symptom would be something like, I have a headache. Well, you can't really measure or observe that. You have to rely on a patient telling you that, or I'm tired, I'm so fatigued. Again, that's something that a patient would have to tell you. So that's a symptom. Okay, so when I say that no signs or symptoms, that means that people are not experiencing anything out of the ordinary. They feel healthy and they don't feel like they're affected by the infection. For other people, on the other hand, they may experience watery, malodorous diarrhea. Malodorous means this is beyond smelly diarrhea. This is really, really something is wrong. Something has died. Bad smelling diarrhea. They may experience intestinal cramps. The watery diarrhea may alternate with soft, greasy stools. The patient may experience bloating, fatigue, gas. They may have a lot of nausea and weight loss. What the protozoan does is when it's bound to the wall of the duodenum or the duodenum and the ileum, it slows down peristalsis and causes malabsorption. So food begins to actually rot in the gut, and that's the source of the odors in the diarrhea. This particular infection is diagnosed by finding organisms in the stool. This is that stool ONP that I mentioned a couple of slides ago. So what does the parasite look like? Basically looks like this. It looks like a smiley face. So this is the sucker right here, the adhesive disc. And right where it crosses kind of the midline of the organism, there's like it looks like a little smile. Okay, and you can see this better when the picture is larger, but there's like a little smile there. Then the macronuclei for the cell look like eyes, and the flagella for the organism look like hairs. So it looks a little bit like a face, and that makes this particular parasite really easy to recognize on a slide. So that's how people tend to diagnose it, symptoms and this image. This is a picture of what the cyst form looks like. Next we have trichomoniasis. It's also sometimes called trich. Trichomoniasis is the most common curable STD in the United States. There are about 8 million new cases every year, but only about 30% of infected people actually develop symptoms, so most people who are infected don't even know it. Both men and women can be infected, but the disease is more common in women. All right, so trichomoniasis is caused by a protozoan, a parasitic protozoan called Trichomonas. vaginalis. And there is no cyst form for this protozoan. So when someone is experiencing symptoms and they go to the doctor about it, the doctor actually has to recover trophozoites in order to diagnose this. So in women, the vulva, vagina, cervix, or urethra can serve as a site of infection. For men, the urethra is the part that's usually infected, but infections can occur in the prostate as well. Symptoms in males include itching or irritation inside of the penis, burning after urination or ejaculation, or discharge coming from the penis. For women or females, symptoms include itching, burning, redness, or soreness. There can be severe inflammation of the vagina. In fact, the vagina can become so red and inflamed that it gets the nickname strawberry vagina. There can also be severe inflammation of the vulva and cervix. Females often know that they have some kind of infection because their discharge changes. Either the amount changes, usually it's an increase, the viscosity changes, maybe it becomes thinner, for example, the color changes, and then there's an unusual fishy smell that usually occurs with the discharge as well. Transmission of this particular disease happens typically during sex, where the protozoan moves from the penis to the vagina, or vice versa. People can also become infected through indirect contact with trophozoites. So, for example, if during sexual activity, implements, toys, things like that are used, then that would be indirect contact. Remember, in indirect contact, you don't come in contact directly with the infectious organism, but rather there's some non-living thing that's part of the path of transmission. Next, we have amoebiasis or amoebic dysentery. This is a disease that occurs worldwide, but it's more common in developing countries. The prevalence of amoebiasis here in the U.S. is only about 4%, and cases in the U.S. usually occur in immigrants who have recently arrived in this country. Most people will be asymptomatic. However, that said, those that experience symptoms may have dysentery. So dysentery is inflammation of the intestines, and it usually results in severe diarrhea accompanied by mucus and blood. There are two organisms that are responsible for this disease. You have Entamoeba histolytica. Histo means tissue and lytic means destroyer, so it's the tissue destroyer, and Entamoeba dispar. These protozoa are acquired via the fecal-oral route most commonly, but they can also be acquired by eating fresh fruits and veggies that have been washed in water that's contaminated with cysts, or drinking water that's contaminated with cysts. All right, so I already said that most people with these infections are asymptomatic. Sometimes people have dysentery as a result. Occasionally, people who are either very young, maybe people who are pregnant, they have a weak immune system, or they're malnourished, maybe they're travelers, again, maybe they're recent immigrants, or maybe they're institutionalized, they may be a little more predisposed to getting the infection and experiencing complications. If amoebiasis is not treated, then peritonitis can occur when the protozoan burrows into the intestinal wall. So ordinarily it's in the lumen of the intestine, but it can attach to the wall and start to burrow through. Okay, so that can lead to peritonitis. If the protozoan leaves the intestinal wall, perforations may occur. Uh, the If the protozoan makes it to the bloodstream, it can travel to other parts of the body, including the liver, where it can cause amoebic abscesses. Could also travel to the lung area, where it can cause pleuropulmonary abscesses, or it could travel to the brain, where it can cause brain abscesses. So now I'd like to talk about a disease that is here in our area of the United States. This is called babesiosis. Sometimes it's called Nantucket fever. And this is a malaria-like infection. In fact, I've even heard babesiosis called the malaria of New England. This disease is caused by Babesia microti, and this particular protozoan is an apicomplexan. So your apicomplexans are protozoa that have very complicated life cycles that involve multiple hosts. So let me give you an example. You looked at the life cycle for malaria. Well, malaria is an apicomplexan. You'll remember that malaria has a mosquito host as well as a human host. Babesia has a mouse host, a tick host. Humans can sometimes be hosts, and occasionally cows as well. I've put a video in your lecture folder that reviews the relationship between mice, ticks, and this protozoan. Suffice it to say, it's the ticks that are going to transmit the disease to humans. These ticks, by the way, are those same deer ticks that transmit Lyme disease. So it's possible that someone can be co-infected with Lyme disease and Babesia at the same time. In addition to the ticks transmitting this protozoan, it can also be transmitted by blood transfusions. Since about 1979, 150 people have been infected with this protozoan through the blood supply. So I happen to know that in Rhode Island, blood donations are usually tested for the presence of Babesia routinely. So take this next statement with a huge grain of salt, because most experts think that cases of Babesia are underdiagnosed in this area. But here goes anyways. Most cases of babesiosis are asymptomatic. Remember, big grain of salt with that statement. Some people who become infected will experience flu-like symptoms. So they'll have fatigue, for example, fever, chills. And let's think about what's happening at this time. So when someone's experiencing these kind of flu-like symptoms, in the body, the parasite is infecting red blood cells. It infects a red blood cell, it undergoes schizogony, producing lots and lots of parasite. The blood cells rupture, and the newly emerged parasites go on to infect new red blood cells. Schizogony happens again. The red blood cell lyses when the protozoa are mature, and then those protozoa go on to infect new red blood cells, and the cycle happens over and over and over again. So this is why there's fatigue and fever. Remember, your red blood cells are responsible for transporting oxygen, and oxygen is essential for making ATP, aka cellular energy. So if you're not able to transmit oxygen because you've lost a lot of red blood cells, then of course you're going to be tired as a result. Some patients will develop hemolytic anemia as a result, and that leads to hypoxia, so low oxygen levels in the tissues. Other complications can include things like renal failure, as the body tries to get rid of all the damaged and dead red blood cell debris. The liver and spleen may begin to swell and enlarge. This is maybe one of the complications that happens after someone's been infected for an extended period of time. Okay. Another complication that appears to be kind of common is acute respiratory distress syndrome, or ARDS. Yeah, that's right. This is the same condition that many hospitalized patients with COVID-19 have, and some die from. Eventually, ARDS can lead to lung damage. So this is not a trivial disease. Now, these complications tend to be more common in people who are immunocompromised, and about 10 to 20% of immunocompromised individuals who are infected with Babesia, they'll die. Okay, 10 to 20%. Diagnosis of Babesia happens through blood smears. So someone has to visually see the parasite in blood smears. There are PCR tests that can be done, but they're not used routinely. The last protozoan that I want to mention in this video is Toxoplasma gondii. Remember, this is that protozoan that infects cat intestines. And I believe we talked about this way back in our introductory chapter to microbiology, when we were going through all of the different types of microorganisms that are studied in micro. I won't talk about this protozoan right now because I believe we've already covered it, but I will put a video in your lecture videos folder. I think the video is about 6 minutes long that generally summarizes some basic points to remember about this protozoan. So now we've reached the end of the video, and we've talked about some of the basic characteristics of protozoa. We talked, for example, about where they live. We talked about how they're transmitted. We talked a little bit about their reproduction, their motility structures, and we talked about the fact that some are free living, but others are parasites. We talked about trophozoites and cysts and how protozoal infections are acquired and diagnosed. Remember, we talked about eating contaminated food, drinking contaminated water, stool O&Ps, fecal oral route, all that good stuff. Finally, we described some common protozoal diseases here in the United States. And of the things that are listed here in the summary, you will probably need a little bit of everything for your exam number 2. So you do want to go through the material and be familiar with it, since exam number 2 does allow you to use your notes. You want to be familiar with everything, but you don't have to memorize everything yet. On your final exam, there will definitely be some basics about protozoa from this lecture, and you'll need to know that material by the final exam. All right, so that's it for protozoa. Next we'll do the helminths, and then we'll move on to viruses. I'll see you there.